will you come today to?
I'd probably pop in to fill the fridge.
Okay.
But today's holiday, so.
Oh, really?
Yeah.
What?
It's a public holiday.
I didn't know.
Yeah. But I think Valentina is coming in.
Okay. Okay.
But yeah, otherwise it will be very empty at work.
Yeah. It's good that you sent this to me.
So yeah, don't feel obliged to come in at all. Don't feel obliged to come in at all today.
Okay.
Okay. Right, let's wait another minute because a couple more people, here we go. A couple more people wanted to show up today. Oh, incognito. Good morning, Laura.
Hi, good morning.
Hi, there you are. How are you today?
I'm sleeping. I'm still in my PJs.
So no paper, I take it?
No.
Fair enough. Let's see. Might be quite a small group today. Which is also fine. Let's see. Let's just wait one more minute and then we can start. Just want to avoid that. You start a presentation and then people drop in. So they miss the important things for the discussion after. Well, I guess it's their loss. Here we go. Just on time. Good morning. Everyone's incognito today, it's fine. Right, welcome to Neuroccino on this public bank holiday. Thanks for joining us. And we have one paper today, and I'm going to do not a book review, but a book overview. So we see how that goes after the paper, but let's start with Lilith, who has a fantastic paper for us. If you want to go ahead and share your screen, Lilith, and then take it away.
Can you see my screen?
Perfect.
Okay, so yeah, I want to present today a paper that is not recent at all. It was published in 1902 by Kinnaman in the American Journal of Psychology. And the title, as you can see, is Mental Life of Two Macaques Rhesus Monkeys in Captivity. So overall, I think I can divide this paper into big parts. So the first one is more about methodological issues, and the second part is where actually they are doing some experiments on these two cute little guys, Jill and Jack. Yeah, and so in a methodological part, it's just he describes different ways to study animals, and that includes free observation of animals in a natural habitat or in captivity, or using training method, or the method where you are studying a single case, and it's single case by studying a single case, you can reveal some type of universal law. Like, for example, if you're studying one dog, you understand not only his trajectory of this development, but also you understand the nature of dogs in general, as this professor wrote. Yeah, and of course, the last method that he talks about is experimental, and he reminds us about Thorndike, one of the first behaviorist scientists. And of course, he says that in experimental method, you don't have a personal factor here involved, but he points the idea that the method, the test you are choosing, is actually important. The way you are, the type of test you are using, it can determine somehow the results you are getting. So, but at the end, he summarizes all these methods and says that we should not exclude one or two this method. We have to use all of them. They are complement each other. So if we use not only experimental method, we can maybe improve our knowledge in comparative psychology. So then he starts with description of his these two monkeys, and this was a super funny part for me. I never seen this way of description of something in scientific literature. It was really like reading a story before going to bed. Like, for example, he wrote, "When baffled in an attempt to open a box for food, Jack walked away uttering a mild audible sigh." And he's trying to actually verbalize the sound the monkeys were making, and or they have shown no sign of playing in any way. They took life seriously. So the way he describes the monkeys, he was like an artist, and I think this is a cool feature of scientists. When you are trying to capture everything, you don't have this tunnel vision, and you just try to see and describe it verbally, things that you are seeing, and this can help to put forward science. I don't know. I think it's an important feature, and I was amazed by his skills to do this. So later, so in this area, he just was putting some random animals or objects in the cage, and he was observing what is happening, what are the reaction of these two monkeys. And then he starts. So his experimental work was divided in two parts. So the first part is trying to replicate Thorndike's work with monkeys, and then he tries to add some more complex situations and add some new hypotheses and new experimental settings. So like with the Thorndike's work, overall how it works, like you have some type of box, and in the box you have you are putting a foot, and monkey should somehow open this box. Basically, that's it. There are super complicated structures of these boxes, and when monkey succeeds, you again putting this. You put again food inside, and you do this several times, like multiple trials, and then of course. You get this famous learning curve when initially it takes too long for a monkey to understand how to open the box, and then at some point monkey reaches plateau. So it opens the same box faster than it was doing initially. And then what happens? And so as Thorndike, Kinnaman, he interpreted this result as in a classic behaviorist manner, where we observe in monkeys gradual learning by gradual elimination of unsuccessful movements, and the gradual reinforcement of the successful one. So basically, it's like trial and a happy accident method, where only important and useful movements are staying, and other type of movements are just fading away because they are not getting any reinforcement. And then he starts doing his experiments, and he adds more complex ones. And this here, it's interesting because he gives these boxes not only to monkeys, but also to adults and children. And he tries to also see how they behave. And honestly, I was thinking that he's not going to say anything about this, but here he actually wrote by himself that the nature of the locks of these boxes gives little opportunity for reasoning. So here is, like, I think, a problem, because he's giving us some type of setting that even for humans it's not impossible to show any reasoning. So it means that the test that he chose, he actually determines the results already. So I don't know, I like watching some videos with the puzzle boxes that could be super expensive and really complicated. But overall, they can be divided, I think, in two types. The first one is when you're trying to random stuff with a box, because you cannot, there is no opportunity to understand the hidden mechanism of this box. So you try different random things. And the other one is when you actually try to turn on your logic, and you are getting to the you are opening the box by using your reasoning, your logic. So the way the box is constructed gives you the specific type of result, and he by himself writes this. So yeah, maybe that's why, like, for example, another scientist from like Gestalt framework with a monkey, for example, Kohler, he had actually also, he was experimenting with monkeys, right? And in that case, the solution the structure of the task was made in a way where a monkey can show his way, his reasoning. And in behaviorist approaches, there is no such opportunity. So that's why they are interpreting their results, it seems, in this trial and error manner. Yeah. So yeah, and then he tries different other interesting experimental settings when he tries to understand if monkeys can discriminate different shapes, if a monkey can differentiate different size of objects, colors, and yeah, somewhere there were also colors. And here he and here what was interesting is that monkeys had this association not with a specific color, but they did some type of generalization. So they were choosing boxes where were lighter than the previous one. So there was association not with a specific red or green color, but somehow they understand this ratio between the brightness between two different colors, and they were using only lighter colors. So it is, and here he starts to talk about of generalization of a low order, and I don't know. I think it is also an interesting part that maybe is not possible to explain from behaviorist point of view. I don't know. And yeah, and today morning I found out that actually this is not the end of the story. It's only the first part, and there was a second part of like also another 50 pages, but I haven't read. But overall, he does, as I understand, other tests with understanding if monkeys can perceive numbers, like calculate some stuff. And he does other interesting experiments with monkeys, with the mazes, and also he has even a chapter about individual differences of monkeys, and he does something about smell. Um, yeah, and it describes the individual differences of two monkeys. And interestingly, he says like the individual differences are apparently as great as they might be expected to be in two human beings selected at random. So this reminded me one recent paper, I think 2017 or 18, where it wasn't about individual differences of behavior, but it was about individual differences of brain. And there was actually this idea that monkeys are has as much structural difference in brain as a human being. And this is cool to find some seeds of this idea even in the paper of 1902. So yeah, this basically it. He also interesting chapter about general notion about this generalization. And he discusses if monkeys has this ability to do abstractions, more higher order cognitive functioning processes. So yeah, overall, I really enjoyed this paper or book, I don't know, but I think it is interesting to read the old ones as well, because you see this naive way of describing experiments, and sometimes you get really bright ideas just in one sentence somewhere written, and then you see some type of link with the modern papers, and this excites me. So yes, thank you. If you have any questions, I can try to answer, but I don't know all the details of this paper.
Thank you so much, Lilith. It's great. We didn't have a historical paper for a while, so I greatly enjoyed it. And I can see what you mean with it's more reading like a novel than a scientific paper. There's also a lot of tangents in the description. Like when one of the experiments failed because a friend came by the lab and they chatted rather than observing the monkey. You would not.
This part is really amazing, where he described general observations. It's just like you're reading a book.
Yeah, you would not find that in today's papers.
Yeah.
Right. Are there any questions? Go for it.
Yes. Thanks for the presentation of the paper. It's really interesting, and yeah, about how the science is described and done. Don't you think I haven't read a lot of very old papers, but this paper seems to describe things in a more laid back way. So don't you think that it could be a good thing to add a little bit, like to be a little bit more relaxed in the way we describe science? Like, I mean, some papers are so much about only facts that they basically forget the reasoning. They forget the story, not the story in a like telltale way, but the story in the way we came with our Christians and how we insert them. There is a path, and sometimes they skip all the parts, like all the failed experiments. So that's a big. I think that's the community agrees that it's a big problem that failed experiments don't make it to the publication. I mean, it's changing, but there's still a long way. So yeah, do you think the science was maybe described in a more continuous way before, and that make it maybe a little bit easier to understand and even to reproduce, maybe?
Yeah, I think no, I really agree that sometimes you see the methods part that covers all the paper, and I think it is good because it is the way how you can replicate the same study. Because if you describe your experiment or the way you are analyzing data, then no one will be able to replicate it, and so it is not that good. But at the same times, yes, like introduction part, you would like to have some also historical. For me, it is interesting when person writes about historical papers and somehow tries to link it with the modern papers. But I think papers have some limitation in journals where you have to fit in some type of format, and maybe the good compromise is that these supplementary materials, that you can put all these very, very detailed information in supplementary materials. And if a person wants to replicate it, just look to these supplementary materials. And yeah, but yeah, I agree that sometimes I want more like meat part of the study to understand the question, the problem that they are trying to solve. So yes.
Yeah, that's a good idea with the supplementary material. Maybe also it could be a good way to present the failed experiments and failed methods. Um, yeah, because I also sometimes reproduce the same errors that a thousand scientists made, but it was not reported, so we had no idea it was an error.
Yeah, it's this confirmation bias,
Yeah.
right, that exists. Yeah, and yeah, I think this supplementary thing can help, even if they can write, okay, we tried this method and it didn't work, and it already saves time for other scientists not to
Yeah.
repeat the same mistakes. So yeah.
I'm playing devil's advocate here, and obviously it's not our belief, given that we read papers every week and share them with the community. But do you think that having longer papers or extra supplementary material or telling the stories behind papers is something that people will actually read in a time where we're heading for shorter and shorter sound bites?
I like, you mean that people will not read the introduction part if it will be super long? That's what. Yeah. So for example, also there is another extremity, I think, when you are starting to read the paper and you already know the first paragraph what it will be about. So sometimes every time you have this introduction, if you are, for example, a proficient in some type of field, you start the paper and you see like, okay, this person starts again with the same introduction, and you just skip it. I don't know what to do with it, but as a person who who goes like to new areas now, it is for me kind of rewarding when I start to reading the introduction and I understand, oh, okay, I already know this stuff. But I imagine how could it be boring for people who are already studying this area like for lots of time. So I don't know, but I feel like it is still important to just shortly say and introduce all the problems and questions, also from historical point of view. I don't know, for me it is more it allows me to complete the story of these multiple types of papers in modern science. So
again, devil's advocate, we could argue that we have reviews papers and books for that reason. So we don't need to make every scientific paper longer.
Yeah,
because we have complementary.
yeah, you can. People are citing the reviews, and it is helpful actually. But still, maybe like some short, compact way to say, okay, there is this problem, and if you want to read more about it, here's the review. And people are doing that, and I think this is the way. But still, I think we have to adding more information in supplementary material, and still do not forgetting about the reason why you are doing this research by linking it with some problems that have been studied like for decades and so on.
But I agree, as a reviewer of papers and editor for journals, I get increasingly frustrated these days when I just see fancy methods thrown together just because we can, without the explanation of the theory behind it, why this is actually a good strategy and why this is needed, beyond the fact that it's cool to do it. So that's definitely something I think that we need to bring back, have more theory and more experimental thinking.
Yeah, and
why we do B to get A, for example.
Yeah, and especially like in this paper also, we can see this naive form of the test that you are using determines your results. So you have to really explain why you are using this method or another one, because it is highly probable that already your results are somehow, in some sense, are already determined.
But it's interesting that they explained that they made an experiment, and then they tried to confirm that the experiment was correct. That's what they did, right? And then they explained, oh, actually the experiment is biased towards one kind of result, so we can't really explain much with these kind of things. And I feel that's something lacking in a lot of science in general, because sometimes we find a cool experiment, and maybe it's just because we are pushed to publish a lot, but sometimes you won't describe, or you won't even have the time to do the work to see if your experiment is actually good. And yeah, that's very interesting that they do that. They criticize their own experiment, and that helps to understand the reasoning, the whole pipeline of the research.
Yeah, for example, with this idea of colors, that they are not specifically associated with a specific color, but there is some type of generalization which is maybe not possible to explain from behaviorist point of view. But still, like the last chapter of this book, I don't know, it finishes with this discussion of general notion of different types of ideas. And this is really cool that they didn't try to like explain everything from their point of view, but they tried actually to develop and say, okay, maybe we're kind of wrong, or they're trying to explain it from their point of view, but still they are trying to question something. And this is also nice when you see it in these old papers. And you can see also the development that later maybe these Gestalt psychologists are coming, and they are putting more effort and focus on facts that people from behaviorists were not able to explain it as good as they can, for example. And this is nice, yeah.
Do you have any idea how long those experiments were going on for? It feels like this is a considerable amount of time he's talking about in the paper.
I don't know. I don't know, but this is really huge like they are studying smell, I don't know, colors, there are multiple trials. I don't know how long it was, but this is a great work, really. Even if it's super simple in description sometimes in comparison with the nowadays descriptions, but it is really, you are studying a monkey from different perspectives, including just observation and including like studying different separate modalities with a very interesting experimental settings. And I don't know. And they had like just 2 monkeys, female and male, and still they are also describing individual differences. So it's like covering all angles of studying monkeys maybe. And I don't know, the only thing that is missing maybe is observation in a general, in the natural habitat, not in captivity, but still, yeah, it is amazing work, I think.
Interesting. Nowadays, where everyone needs many papers to make it, you would probably break that work down into 10 individual papers rather than one 50-page long.
I think at least 10, because it's a lot, like each experiment. And then no, I think it's, and then there is also methodological, also a bit of philosophy. So it's like, I don't know how many papers could have been from this one.
Great. Well, thanks for reading part 1. Looking forward to part 2.
No, but in the second part, it's almost more or less the same, but yeah, different modalities, just, yeah.
No, it's great to have those historical papers and see the difference as well. I was also thinking if part of it is just zeitgeist, because like different countries and different times have this philosophy about how to write scientific papers. And some think you have to make it really complicated to show that it's an academic exercise and that is like an elite job to do, whereas others argue that the writing should be very clear and understandable, especially in the framework of being publicly funded, giving back to society. It has to be written in a manner that people without an academic background can also at least get the basics of it. Mm-hmm. So there's very, very different schools of thought about how to write. And then obviously our methods have come a long way as well. So they are just way more complex these days, which is harder to...
It was also nice to see that they are actually writing the size of the box, like it matters a lot. Yeah, this is nice when they know how to describe at least something more strictly, and they are showing that we can do that. We can measure the size of the box and we will put this in the paper. Yeah.
No, it's great. It's great. I like it. It's also that, like, no matter how fancy our tools are and our methods or our experimental design, we're always trying to answer a simple question in the end. Mm-hmm. Like the questions itself are usually basic, and then we layer methods on top of it, which makes it super complicated. And sometimes it's easy to get lost in this complex methods world and forget what the actual fundamental question was.
Yeah, and you can find these fundamental questions in these old papers in a more open, naive way, and that's why it is interesting to read this kind of stuff, I think. Yeah.
I think reading historical papers is massively undervalued and something we should be doing on a regular basis. It's quite, and as you said, you get fantastic ideas, or you realize that something you think you just discovered was actually already there. So this hurts a little, but
no, it's just seeds. It's not like it isn't described like in all this complexity, just small seeds, and it is cool to find them.
Great. Any other questions? No? Okay, in that case, thank you so much, Lilith. That was great. And I'm not going to keep you too long. I just thought that I briefly talk about references, because the question comes up a lot, especially doing teaching, what kind of books one should read to learn about the brain, learn anatomy. So I thought I'd just share a couple of insights and books. And as Michel is not joining us today, I can actually present his book. So hang on. So the first first thing I would recommend is get a brain. There's nothing like studying the real brain. If you can't get to a morgue, get a model. This is one that I 3D printed myself. I have a template on my website that you're welcome to just download. It's my own brain. And you can just get it printed off wherever you want. So you can have a model at home. And it really helps to appreciate the 3D structure, makes it a lot easier. But in terms of learning what those structures are, let me start. I'm going to go chronological with the books that I learned. So this is the first one. It's called Photoatlas Neuroanatomy, which is basically a picture book of neuroanatomy. And I'm showing my age now, but it comes with a DVD at the end. You can probably get an updated version of the book now and just download the video, but it's super, super cool. And this is a German version, but really the terms are all Latin, so it doesn't really matter. You can see I used it quite a bit. And what is good about it is that the size of the book is fairly small, so you can actually take it to the morgue when you do your actual dissections. And whenever I hit a wall and anatomy wasn't good enough, I added my own little descriptions of what we can see. And I'm sorry, I don't have a fancy PDF for you like the pros who usually present here, but let me just find a nice, there we go. This is a picture you can show in the morning on YouTube. So here we go. So this is what it looks like. You have the structures of the brain, and then you have the descriptions here of what each structure is. And you have that for the gray matter. As I said, whenever I felt like I needed more info, I just put my own little thing in there from papers. And the nice thing is it goes through the sections. You have the lateral surface as well. There's a couple of pages you want to skip because it's the anatomy of the full head, which is a bit not breakfast friendly, let's put it that way. But you also get cross sections of the brain. So you can study the subcortical structures, the nuclei, the deep gray matter. And then there is a couple of, here we go, Klingler dissections where you see the white matter. So this is really a lovely, lovely book to get started. And it has helped me a great deal throughout my career. Then the next book, and you're going to laugh at me, but this is a human coloring book, human brain coloring book. And it's basically a coloring book for adults, which sounds ridiculous to some people, but part of my anatomy education actually included someone asking me if I know certain structures. I'm like, of course I know them. And then I was asked to draw them. I was like, maybe I don't know them as well as I thought I do. I can label them when they're in front of me, but I can't draw them myself. So I actually spent quite a lot of time going back and using this book to draw. And it's a fantastic way to learn your anatomy that way. And it's also written by one of my personal heroes, Marion Diamond. Here you go. So you start from the surface and you just work your way through. There's also different networks of how they link and connect and make your body work. And you always have descriptions on the other side as well. So yeah, it's a fantastic resource. This is actually one of the hardest questions to ask in a master's exam. Those people are super highly trained, but when you just ask them to draw a neuron with all the parts, you'd be surprised. So this is a fantastic book, the Human Brain Coloring Book from Marion Diamond. Right, so these are two books. And then I said I'm going to use the opportunity with Michel not being here to present his own book. So this is the Atlas of Human Brain Connections from Marco Catani and Michel Thibault-Deschâtel. And we usually have a rule that we don't present our own work during Neuroccino. But the reason I'm presenting this briefly It's because it is actually a fantastic reference for the white matter. Can you see my screen? Yes, great. So this book begins with historical background, generally on the white matter and the anatomy of the brain, the different types of anatomy we can study. And then let me just fast forward. You have all the labeling of the brain, the surface, the ventral surface, the different lobes, it's all in there. You have historical drawings and the people and the story behind it. And then we carry on. So I'm just scrolling through it. And then it is obviously a DTI atlas. So you have the method of DTI, how it's done, what you can do with it, the history of the white matter as well. Here we go. And then you have those beautiful pictures in there as well from historical books. There we go. A couple of famous people in the field. The different things you can do, you can dissect out, you can segment, you can plot indices along the tract, you can use it to parcellate the cortex, all the different things that can be done with tractography. Then you have chapters on the theory and the models of the brain and how the brain might work, from holistic to localizationist to associationist. And then the different models put into perspective and how we can study it with tractography, the lesion method and tractography, inter-individual variability, moving from a topological to a hodological approach. So from individual brain cortical lesions to white matter disconnections. Then the individual lobes, here we go, parietal, frontal, occipital, and temporal. But then the most useful thing is that you actually have an atlas of the white matter slice by slice through the MNI. And here in color, you see the DTI reconstruction of the tracts where they are located in the MNI brain. And obviously this is also available as a digital atlas. It's actually also implemented in the latest version of MRI Cron. So you can just open the maps and look at it. But it's quite useful as a reference to see where you are and where your white matter is. And you have that for all axes. So here we have axial. You also have the coronal slices. Here we go. And we also have sagittal slices in there. Here we go. This is what it looks like on the sagittal. Now, in addition to that, what is super, super useful, oh, and then you have a clinical chapter for each individual tract, what the tract correlates with, left-right hemispheric differences, etc. Clinical. Where is it? Clinical part is a big part of the book. It's not in here. Yeah, so it's a great resource you can use. Obviously, it's from 2012, so the DTI algorithm did not cover the full brain, but there's new atlases that now do cover the full brain. So this is just to give you some pointers of where to start. And for those of you who are interested in more history, given that we had a historical book today, this is the advantage of working in your library. I can totally recommend this one, The Origins of Neuroscience. Stanley Finger. It's a fantastic resource where current models and ideas came from. who the people behind them are, what the controversy at the time was. There's, for example, a big chapter on language and Broca's area, and if the name Broca's area is actually legit or if it should be someone else's cortical area. It's a lovely, lovely book, and it reads a bit like the paper you just presented, as in it's an easy and interesting read, but super, super exciting. That's about the books I wanted to recommend to get you started. As you can see, there's many, many more. And I'm happy to go through them one by one throughout the years. But this is just an overview. So these are good references to get started on anatomy, get started on a history of neuroscience. And then we're going to present many more books on current models and methods as well. Any questions? It's not really a question topic, is it? Right, okay, in that case, thanks for joining us this morning. I also wanted to alert you to the fact that we have a talk this week, and that will be on Thursday, 4 p.m. Paris time. I was going to say Paris time. And we have a talk about hedonism. Hang on, let me just quickly show you the slide. Tuck, here we go. Here we go. That's the talk this Thursday at 4 p.m. And you're all welcome to join. The way it works is we have all the links up here. Just come to our website. And as always, you can join us live on Zoom, or you can watch it live on YouTube or watch the recording afterwards. But we're super excited about this one. So this is this week, Thursday, and you're all welcome to join. That is it for today. Thank you so much. See you Thursday and see you again next Monday for the next Neuroccino. Have an exciting week in science. Bye.